I want to thank you all for making the effort to be here. I appreciate the extra schlep, and I admire your ingenuity to actually find the room. So thank you very much. Before we start on the next chapter, can we maybe adjust so we don't have quite the ring? Thank you. Or maybe I do that. Let's see. Maybe that'll be better. Before we get into the current chapter, I'd like to know if there are any other questions about the organization of the test on Friday. Again, as on the first two tests, we'll ask people to wait outside if you haven't been able to get there by 3:30, and then we'll admit you at 3:40. If at all possible, try to have ID so we can check the little boxes, and don't panic. I apologize for the light level. It's probably not optimum, but the next up light level makes the screen vanish. So this is what we're going to have to live with, guys. I hope I can be scintillating enough to keep it all going. I noticed looking at the syllabus that this chapter initially had 2 days devoted to it in the syllabus. And I think what happened is we took 2 days on oxidative phosphorylation instead. So rather than getting further and further behind, I'm going to focus on 1 day on this. We may not cover all the material in the handout. So whatever we do get to cover today, that's what we'll cover. And if people want to hear the rest of it, just sort of for the fun of it, I'm happy enough to talk about that. All right. We've spent quite a bit of time so far looking at the organization of metabolism as pathways. We've looked at how things can be transforming energy into more usable forms or storable forms. We've looked at how you can move nitrogens around and so on. But the challenge remains to make all of that balance appropriately. If all you are is an amoeba, then balancing the pathways is no problem. They're all in contact with each other. There's no real integration that's needed. You're already one cell. But as you get bigger, it gets more and more necessary to find some way to make all the different parts of the organism talk to each other so that the right pathways are going in the right organ at the right time. And so this integration of metabolism, for one thing, makes sure that the needs of each tissue are met, and that's done without wasting energy, without wasting materials. You're going to see compartmentation playing a big role, that by having metabolic pathways either in different parts of the cell or in different parts of the organism, it becomes possible to regulate things differentially. We're going to touch a little bit on hormone mechanisms and signaling. Those of you who've taken endocrinology will know that for me to try to give that in less than a lecture is obviously a skim, but it'll be a taste, a tantalizer. We'll look at least 2 of these hormones, insulin and glucagon, because they play such central roles. And if we have time, we'll talk a bit about diabetes, which is a disease of glucose and of fat metabolism. And we may get to that. We'll see. This is a dramatic demonstration, I think, of just how compartmented the various pathways are. And so you can see that the matrix of the mitochondrion is substantially involved with things that involve burning things up. So you have the citric acid cycle, which is used partly for burning things up. You have beta oxidation. Ketogenesis is making something, but it's making something you can export to burn up somewhere else. Amino acid degradation and the urea cycle. A little bit of synthesis, so amino acid synthesis occurs here, and some things get withdrawn there, but a good deal involved with burning things up. And then in the cytoplasm, you have pathways that are synthetic. So you have gluconeogenesis, fatty acid synthesis, nucleotide synthesis, some more of the amino acid synthesis, and parts of the urea cycle. Glycolysis, of course, is involved in breaking things down. That's such an ancient pathway that it was in the cytoplasm before cells were eukaryotes. And then these various transporters are just involved in making sure that these 2 families of pathways can communicate the materials that are needed to get things back and forth. Just an example, the carnitine acyl carnitine transporter to get acyl tails inside so they can be oxidized. Citrulline transporter getting things back and forth between the urea cycle. But beyond that kind of compartmentation, which is true for any eukaryotic cell, we also see compartmentation at the level of the organs in organisms that have that. And this is one example of how that can work. You're all familiar with the idea that in anaerobic exercise, the use of ATP is exceeding the rate at which mitochondria can make more ATP. That you're compelled to produce your ATP by inefficient means like glycolysis, which end up producing a lactic acidosis or something like that. So the pyruvate that gets produced at the end of the glycolysis, when it's converted to lactate, that restores the NAD plus that's needed there, but you have to take care of the lactate then. And what happens is the muscles are going to be the ones that are extremely active in anaerobic exercise, and they will release that lactate into the bloodstream. The bloodstream will carry it to the liver, and the liver, of course, is the major home of gluconeogenesis. And so in the liver, at its leisure, the liver can use fatty acids or whatever to regenerate glucose molecules and shuttle it back in there. And so the net effect of this shuttling between the tissues is you're moving energy from the liver to the muscles. And you may remember my example of the alligator that can leap and build up a lactic acid debt. It takes hours to pay off. I apologize for my voice. Am I clear in the back? Is this reaching back there? Okay. It sounds foggy to me. Another example is when either extremely vigorous exercise, so if you're doing triathlons, if you're doing ultramarathons, or if you are actually fasting beyond a certain point, then the body will have to start breaking down some of the muscle protein in order to generate carbon backbones and so on. And in situations like that, now you end up having more nitrogen than you need on a daily basis. And so what happens then? Let's take a look over here in the muscle. So the glucose goes to pyruvate as we expect. We can take an alpha amino acid and pass its amine group onto the pyruvate by transamination. So now we have an alpha keto acid, which is probably a fine candidate for the citric acid cycle or whatever. And that may turn the pyruvate into an alanine. Now the alanine can go in the bloodstream to the liver. The liver has extensive capacity for the urea cycle. In the liver, that nitrogen in the alanine will be turned into urea that way, releasing a new pyruvate. And of course, pyruvate could be used for gluconeogenesis, which goes back and can be recycled again and again. And so the net effect of this cycle is to move nitrogen from the muscle to the liver. So example of how these pathways located in different tissues now are working to cooperate. So how do you make the different tissues that can't see each other or touch each other know what's the right thing to do at the right time? And that we can attribute to hormones. Hormones come from the Greek word that means I urge on. And hormones urge tissues to do things. The general definition of a hormone is a chemical that comes from one place and acts somewhere else. And because it's carrying information, it's integrating the two situations. But hormones themselves do nothing. A hormone by itself is like having a book, but no one can read it. You have to read the meaning of the hormone, and that's going to be happening through signal transduction. And this is a sort of a generic, one-size-fits-all example of what kinds of things happen in signal transduction. And I'm going to take you through it a little bit to see what the general features are, and then we'll see how the specific features are different in different pathways. The ligand in this case is the hormone of whatever kind. The receptor is typically going to be a transmembrane protein. It has to pick up the ligand outside. It has to talk to the inside. So typically it's a transmembrane protein. And it'll typically have a ligand binding domain. When the ligand occupies the ligand binding site, something mysterious happens. And what exactly happens will depend on the receptor. But something happens so that the inside end of this is doing something different now. And that will be picked up and detected by intracellular components somehow. And we'll see some of those somehow. One consequence of this is typically that a second messenger is released, and that second messenger then alters things. Another is sometimes the product here is going to directly target proteins and have things happen. Some of these activities will stay in the cytoplasm, being enzymatic activities. Some of these things will go through the nuclear membrane and actually interact with the DNA in order to have changes in gene expression. We're going to see that in many cases these are cascades. That is, there's something where one step amplifies the signal so it's bigger. And if you have several steps of amplification, then something that starts out as one little hormone molecule can be 1,000 something else when it gets over here. So the second messenger is a common feature. There's always an off signal. We don't have the off signal in this diagram, but trust me, there's an off signal. You couldn't have life going on if you couldn't turn hormones off. And that's a general principle, that degradation has to balance synthesis. Degradation just isn't as sexy as synthesis, but it's just as important. Well, I think I'm kidding. People thought for years that the only thing to think about was mRNA production. Well, if you made it and never broke it down, we'd be up to our eyeballs in mRNA. So obviously mRNA degradation has to happen, and it turns out to be every bit as regulated as the synthesis. So, but it's not immediately intriguing. All right, back to biochemistry. One of the major players in signal transduction are protein kinases. So a protein kinase will be something that puts a phosphate group onto a protein. And there are two big families of protein kinases that we'll see many times over and over again in signal transduction. One of these families targets a tyrosine, and so this is a phosphotyrosine. You can see the phosphate now. That would have been a plain old tyrosine without it. Or it'll target a serine, or it'll target a threonine. These two are lumped together in a family because serine and threonine are similar enough that this family evolved as a group. Excuse me. So this is what phosphoserine would look like. This is phosphothreonine. Now all three of these, you notice, the two families, what they do is they turn a neutral residue into a charged residue, into a very charged residue. So they have the potential to significantly alter conformation or protein activity. The body has to be able to budget its energy. If we don't budget right, we get into trouble with the bank. Body doesn't budget right, you drop dead. A little more important. And so the body has worked out mechanisms to use the most appropriate fuel at the most appropriate time. So for instance, right after you've had a meal, say an hour or two after you've had a meal, 50% of your energy will be coming from carbohydrate, about a third from fatty acids, about 17% from amino acids, even at that time. If, on the other hand, you don't have any snacks between your 6 o'clock dinner and your breakfast the next morning at 7 or 8 or 6, that would be an overnight fast, 10 to 12 hour fast. That's enough time to make a significant shift in how the body utilizes fuels. That in that morning, you're now drawing only 12% of your energy from carbohydrates and 70% of your energy from fatty acids. So that window of time, which is, we all live through every night, is enough for the hormones to kind of retool it from SUVs to sports convertibles or something. Amino acids have stayed fairly similar though. A long fast, like a 40-day fast, this is the kind of fast that Gandhi, for instance, would do. At that point, all of your energy is coming, basically all of your energy is coming from fatty acids. This 5% that's coming from the amino acids is just to give a certain number of backbones. And at this point, this is body mass being broken down to provide those amino acids. So an extraordinarily flexible approach to what fuels are going to be used when. In order to coordinate all of this, we have insulin. This is a picture of insulin. It's a small protein, a peptide hormone. And it's a signal in the body that fuel is abundant and do something sensible with that fuel. So for example, insulin in muscle and some other tissues will increase glucose transport. it will increase glycogen synthesis. So these are things that will tend to clear glucose out of the bloodstream and store it for future use. It will turn off the enzymes that can catalyze glycogen breakdown. So it's preventing a futile cycle from occurring. If we look at adipocytes, rather different activities here. Adipocytes are fat storage cells. The effect of insulin here is to stimulate the extracellular lipoprotein lipase. This is the lipase that can sort of vacuum fatty acyl groups out of the LDLs, and it'll stimulate acyl-CoA carboxylase. So that's going to help with the synthesis process of synthesizing fatty acids and triacylglycerides. It'll suppress fatty acid release. So the presence of insulin, which we think of as a carbohydrate hormone, will suppress fatty acid release because it doesn't make any sense to regulate carbohydrate metabolism if you don't also regulate fatty acid metabolism. And in fact, insulin does both. And as I said, the overall message of insulin is that fuel's abundant, store, don't mobilize. There are, of course, many, many more results of insulin, but these are some highlights. Sure.
When you're talking about the way your metabolic feels vary with state,
Mm-hmm.
so what about like right before, like between breakfast and lunch, like you get towards lunch, you feel like
Mm-hmm.
you feel your sugar levels
Mm-hmm.
going down. Like how is that changing your metabolic efficiency?
That's a very good question. So she's asking what happens in a shorter fast between, say, breakfast and lunch. And the short answer is it depends on what you had for breakfast. If you notice, let's go back here a second. Whoop. So here we are, overnight fast. You're turning off your carbohydrate metabolism. You're doing a lot of fatty acid metabolism. If you provide protein in that first meal of breakfast, you're giving carbon backbones to allow gluconeogenesis to happen. If it's a pure carbohydrate breakfast, it's going to go up and come down like a stick, up like a rocket, down like a stick. And we'll see some of these slides if we have time later on in the handout, where the content of the diet will affect how the blood sugar changes. Short answer is, sugar-free yogurt good, eggs good, bagel with jam, not so good. At least not by 11 in the morning. So where is it coming from? Insulin comes from little clusters of cells in the pancreas. And because they look like little islands when you cut through them, they call them islet cells. There's two kinds of islets. These come from the pancreatic beta islet cells. And they are going to be translating, in order to regulate the metabolism of glucose, they have to have an intracellular signal. And this is a rather cute example of how that works. We talked a little bit about these two enzymes earlier, hexokinase capturing a number of hexoses, glucokinase capturing just glucose. This one can be feedback inhibited, this one isn't. And what's interesting here is that the affinity of hexokinase is so much greater than glucokinase that it's essentially going at pretty much full tilt at any cellular glucose concentration you're likely to encounter. It's going as fast as it can, unless you're getting feedback inhibition or something. Glucokinase, by contrast, does not have a very high affinity, and so its slopey region cuts right across the changes in blood glucose that are likely to be significant. So as blood glucose goes up, glucokinase goes up, activity. And so this lets glucokinase be a glucose concentration sensor, and that's part of how the pancreatic cells can work their wonder. I think I mentioned before that glucose is one of the only, if not the only, sigmoidal monomer that somehow it remembers that it recently did it and does things differently. So, insulin by itself does nothing. It's just a little peptide. It has to bind to a receptor, and the insulin receptor is a dimer of dimers, so it's alpha 2 times 2. The alpha regions are outside the cell, and the beta regions are transmembrane, the outer part in contact with the alpha, the inner part inside. When the insulin receptor is occupied by insulin, that will somehow or other communicate through the protein, and the beta subunits then approach each other and now act as kinases by adding phosphate to each other. So they become their own best friends, so to speak. And we find this kinase, it's a tyrosine kinase, and we find this activity on beta chains, also on a molecule called IRS1, insulin receptor substrate 1. So this is another candidate for spreading the message that insulin has come up. We find that many different peptide growth factors can use tyrosine kinases, and that they have many different targets in their target tissues. Peptide growth factors include things like human growth hormone and somatostatin and things like that. Many things that regulate the immune system are peptide growth factors. So what happens? Oh, what happened there? I don't know what they did to me. It worked. Did it work on your handout? I'm just kind of curious. Okay. What can I say? So what are some of the kinds of things that insulin can do to get these effects? Well, I've mentioned already that it has to have a receptor. In both muscle and fat cells, glucose will increase glucose transport, and it uses a cute trick. A cell that can respond this way will have in its cytoplasm preformed inactive glucose channels. When the cell is stimulated by insulin, it can simply bring that vesicle out to the membrane and add those transporters to the membrane very quickly. So this allows for a very rapid increase in the capacity of a cell to transport glucose. Each channel is working the same as it did before. This is increasing the Vmax of the cell because you've got more enzyme. If you get the signal that the insulin's cleared out, you start getting counter signals, then the cell has ways to take these channels out of the membrane and store them back like that. It can also stimulate the fatty acid uptake, as I mentioned, by increasing the lipase activity. So you can sort of vacuum fatty acids out of the LDLs. We also think of another aspect of carbohydrate metabolism is that insulin can tune glycogen metabolism. And we looked early back when we were just starting metabolism at the fact that a few cells like the liver, somewhat the kidney, can make or break down glycogen. And the balance of that, whether there's going to be net synthesis or net degradation, will depend on the relative activity of glycogen synthase and glycogen phosphorylase. We talked a little bit of some of the allosteric regulators back when we were doing that part of it, but I mentioned we were going to see another one later on. The largest changes in glycogen metabolism occur through phosphorylation that are stimulated by kinases under hormonal control. And it's sort of a cute trick because the two halves of the equation have opposite responses to being phosphorylated. So when you have glycogen synthase and you phosphorylate it, you turn it down. When you have glycogen phosphorylase and you phosphorylate it, you turn it up. So you're getting a double whammy. If you're turning one down as you turn the other up, you're getting a bigger change than just one alone. And in the other direction, the same kind of reciprocity. There are phosphatases. Remember I said all signals have an off switch. Phosphatases turn off kinases' effects because they take off phosphates. And so when those phosphatases act, the glycogen phosphorylase gets turned down and the glycogen synthase gets turned back on. So it's a very beautiful way to get reciprocal control and get the maximum effect for one hormone's signal. So that's what happens when times are good. Insulin is the signal that fuels are abundant. Time to store, time to stave away somewhere. Don't use your own stores because, good times are rolling. When you've depleted those fuels, when you've run through most of your glycogen, for instance, then the liver has ways to mobilize glucose in order to feed the rest of the body. And there are several hormones that are involved in this. Glucagon is sugar urger. This is the hormone that urges the sugar out into the body. Epinephrine and norepinephrine you may know as adrenaline and noradrenaline. These are part of the fight or flight reaction. The glucagon is sort of the sister hormone to insulin. It comes from the alpha cells of the pancreas. And its actions are just what you'd expect it to be. The glucagon can stimulate glycogen breakdown. It can stimulate gluconeogenesis, where those amino acid backbones can be made into sugar from fat. And it stimulates the fat cells to go through lipolysis and release fatty acids to the circulation, where they can be used as fuels directly by the muscles, for instance, and they can be used in the liver to do gluconeogenesis. And then epinephrine and norepinephrine stimulate the muscles to use their glycogen stores. The glycogen receptor is an example of a different kind of family of proteins compared to the insulin receptor. It's called the 7-transmembrane domain family, and that's because there are 7 alpha helices that span clear across the membrane. You can imagine that that's the one end of the leaflet and that's the other. Other ones in this family are adrenergic, rhodopsin, and so forth. Just like in the insulin receptor, when the ligand binds, it changes conformation. Unlike the insulin receptor, it is not a tyrosine kinase. Now, some of the ways in which receptors can talk to the cytoplasm is through a family of things called G proteins. And G proteins are called G proteins because they bind GTP or GDP, one of the guanine nucleotides. G proteins can undergo a cycle. So you can have the inactive state where you have an alpha beta gamma trimer that is not active. It still has a GDP bound to it. Some kind of a signal comes that stimulates the release of the GDP and rebinding a GTP. That splits the two parts of it apart from each other, and they're now active, and they'll stay active until this subunit hydrolyzes its GTP into GDP, and then it'll pick up another beta-gamma subunit to do that. This process of losing and picking up is not that fast by itself, and so there are exchange proteins that can help. Guanine nucleotide exchange factor, GTPase activating protein. So you can see that at both of these steps there are factors that can accelerate those reactions. Factors for the factors, so to speak. Big fleas have little fleas on their backs to bite them, and so forth and so on ad infinitum. What are some of the things that G protein is going to do? Well, when the alpha subunit of GT protein is bound with GTP, so alpha GTP, one of the things it often does is to activate an enzyme called adenylyl cyclase. And adenylyl cyclase does what its name says, weird as it sounds. Here's an ATP, and adenylyl cyclase can split off 2 pyrophosphates there and cyclize, like that, the remaining phosphate so it binds back onto the ribose. This has a very different shape than that. And cyclic AMP has very different binding properties from plain old AMP. Cyclic AMP can then bind to a second target, and in this complex, as long as it doesn't have cyclic AMP bound, it's a tetramer, it's inactive, cyclic AMP can bind to the regulatory units and liberate the catalytic units. So now these catalytic units become protein kinases. This family of kinases tends to target serine and threonine. And for example, one target, phosphorylase kinase, would be one of the targets of it. There's always an off switch. Cyclic AMP can be broken down by a phosphodiesterase. So it's right there. It would break that bond. Caffeine is an inhibitor of phosphodiesterase, so one of the reasons caffeine perks you up is it keeps your cyclic AMP around longer. Protein kinase A, and it's called A because it's activated by cyclic AMP, would do different things in fat cells. So in fat cells, it can activate the lipase. It can move the lipase to the fat droplet where it can get more access to the to its substrate. So this is an example of how some of these pathways can work. And the receptor at the top of this, at the place where this, if you want to, there could be a membrane here. The exact receptors that a cell has will determine what signals it can receive. It's like a radio tuned to a particular frequency. The initial steps of this cascade, an active G protein turning on adenylyl cyclase making cyclic AMP activating protein kinase. These parts can be pretty much similar in different cells, even though the hormones up here are different. But the targets can be different. So even though these parts are so similar, in adipocytes it can cause lipolysis because the final target is a lipase. And in muscle cells, it can affect glycogen metabolism because that's its final target. So the protein kinase has many targets, but any one cell will have specific targets, and that's going to be which metabolic activities are going to be modulated. There's another thing to point out here. Adenylyl cyclase is an enzyme, and so it can make many molecules of cyclic AMP. Protein kinase is an enzyme, so it can activate many molecules of hormone-sensitive lipase. And so we've got two steps in here of amplification so that one hormone molecule can turn on many molecules of lipase. And something like that had to be needed. If it was a one-to-one, there wouldn't be any room in our bodies to do anything. There are a lot of pathways that use G proteins. Now that we know the whole human genome, 1% of it consists of G protein linked receptors. So that's a significant fraction of the body. They can do a number of different things. So for instance, some G proteins can inhibit cyclic AMP. Other G proteins can turn on a guanylyl cyclase, so you can have cyclic GMP. Could target kinases and so forth directly. If we look at another hormone, here's epinephrine, it's going to have a slightly different mechanism for amplifying its signal. So epinephrine can stimulate the activity of an enzyme called phospholipase C. And it's called C partly because it is stimulated by calcium. And it will take this molecule, which you instantly recognize as phosphatidylinositol bisphosphate. It's got two phosphates, and that's the inositol. And it will split that molecule up into this, the diacylglycerol, so here's the glycerol, diacylglycerol, and an inositol triphosphate. So it takes a molecule that's kind of like almost a detergent beforehand with a very amphipathic character and splits it up into a very lipid-soluble and a very charged piece. And what good does that do us? Well, something like this. So here, if we look at what epinephrine is doing in a different cell, we're going down. Phospholipase C is activated. It acts on phosphatidylinositol. It makes both a diacylglycerol and the inositol phosphate highly charged. Diacylglycerol can activate the protein kinase C by releasing calcium, so it can go back and activate that. So there's a feedback that can amplify it further. The inositol phosphate can act on the calcium channel to activate the calcium for the protein kinase C. Both the cyclic AMP and the inositol phosphate and the diacylglycerol, these are all examples of second messengers. The first messenger would be the hormone itself. These are the second messengers. It's also interesting to see that binding at one receptor can have branched effects at the bottom. So in other words, when you use the phospholipase C pathway, you produce two kinds of second messengers. You're going to have two separate kinds of effects on the outcomes. And again, since these terminal things are all targets of kinases, the phosphatases are going to be important too. So we have a bit of time to talk about diabetes. I don't know if we'll get through all the material on diabetes, and I will offer an open invitation to anybody who wants to stay if we don't get through it all. Diabetes is a disease of energy metabolism. It's not just a disease of carbohydrate. It's a disease of energy metabolism. But the way that it is first detected usually is that the blood glucose levels get so high that glucose is lost through the urine. And that was detected when this dog had its pancreas removed, and the urine came out sweet enough for flies to come around and sit around the edges of it. There are two major presentations of diabetes. There's one we call type 1, or insulin dependent. We used to call it juvenile. It's an autoimmune disease, principally, that the pancreas gets attacked by the own antibodies. And then type 2, which is insulin independent. So in the insulin dependent form, the autoimmune process has destroyed the pancreatic beta cells, and the body doesn't make insulin. So you need to have insulin from an exterior source. In type 2, the body can still make insulin, but the cells are no longer responding appropriately. It's what we call insulin resistance. We used to call type 2 diabetes adult diabetes until juveniles started getting it. So it's very sad. Now, why does diabetes matter? Well, it ends up affecting basically all the body. If you have this elevated hyperglycemia, a lot of blood glucose, it's happening because the cells can't take up sugar. They're starving inside. The cells, there's wealth on the other side of the membrane, but they can't get it in. And since the body thinks it's starving, it does things like increasing gluconeogenesis, which is just going to make it worse. It can increase serum lipids. It's telling the lipases, put that into the bloodstream. So you see things like elevated LDLs and stuff. Sometimes the levels of sugar get so high that you see spontaneous glycosylation of proteins. That is, glucose is reactive at a slow rate with proteins in a nonspecific way. And in diabetics that have severe enough diabetes, the proteins in the body start just getting sugar added to them nonspecifically. And this usually isn't better for them. It usually makes them worse and tends to progressively denature proteins. Things like a cataract here where the lens proteins, the crystallins, got glycosylated enough to be denatured. It can lead to damage to the nerves. It's very common to see diabetics who have peripheral lack of sensation, and when that happens, it's very easy for them to have damage to their feet. It's one of the leading causes of having to amputate legs. My great-grandfather had his leg amputated for that reason. Another big target of diabetes is kidney damage. You can imagine if you have so much sugar pushing on it that this is not the way kidneys are meant to work. And we see a lot of the vascular and heart disease. Part of the damage to the eyes is glucose being turned into sorbitol in the eyes. But if you eat sorbitol, that's not a problem because it's not carried into the brain. There we go. The major cause of diabetes in our country today is obesity. This is something that has changed dramatically in the last 3 decades. It is playing a major part in heart disease, in cancer, joint diseases. All of these things can be traced back to the increase in obesity and overweight. Correct body weight is a very delicate balance of intake and use. It's, a few calories a day and you start to acquire pounds per year. We don't fully understand how such a delicate balance can be maintained. Most people don't vary by 15 pounds a year. So how do we manage to maintain that? There may be a role for leptin. If you want to read more about that in your book. It's clearly doing something in lipid regulation. We're not quite sure yet. How much of a change? 5% of the population was obese in 1900, 31% in 1988. That's too soon for it to be genes. We still have the same genes. So this is not a genetic shift, it's an environmental shift. And what has changed in the environment? I am being dramatic enough to call it a toxic environment, if you like. Advertising everywhere for food, for alcohol, things with calories. Big shift to processed foods, foods that are really, really yummy and really, really full of sugar and fat and really, really easy to eat. Old days, you wanted an apple pie, you had to take 2 hours and make an apple pie. You know, you're not going to eat an apple pie every day on that ground. If you go to the store and buy an apple pie, you can eat 3. Food subsidies. Because of very effective politicking, and this is Iowa, so I need to be careful how I say this, some states have been able to negotiate tax barriers or food subsidies, the net effect of which is to make the food cheaper than it should be. That is, if it was sold on the open market, that it would either not be grown so much or it would be selling in other markets. Another contributor is the automobile culture. Ever since 1900, there's been this spreading out from city centers to things like suburbs and stuff, which is only possible because of automobiles. And when you're riding an automobile, you're not walking. People in Washington, D.C. and New York have lower rates of obesity because those are 2 cities where you cannot get around by car. And people walk. They walk, they think nothing of walking 15 minutes to get to an appointment. 20 minutes. Happens just like breathing for them, and it builds in enough extra movement. Spectator sports. 1900, what sports was a bunch of you went down to the corner playing field and you played baseball. And now it's the $100 million stadia that you watch on TV while you're drinking beer and eating Twinkies. I mean, this has a real big difference on your calorie balance, This just has a radically different effect on your calorie balance. So, and I think I got your attention on that one. So just how serious is it? 16 million Americans have diabetes. 16 million. You could populate New York twice over with people with diabetes. And at least half of them aren't diagnosed yet. So the effects of the disease are progressing without any effort at control. This is showing how different it has been over time. Some people say, well, we're living longer and it's a disease of old age. Well, it's true when you look across age that a few more people are obese with up to about the 50s or so. But in 1880 to 1900, we're talking about a few percent. And in 1988 to 1994, we're talking about a quarter, nearly a third. And I'm sad to say that this line is higher still again today. 1994 is 13 years away. So this is an overall increase. At every age, there's been an increase in obesity. $1.07 is spent on diabetes. If you think about the things that diabetics need, like strips and more regular checkups and operations for the complications and so forth, you can see why that would be. Cost you roughly 3.5 times as much to provide health care to someone with diabetes. $85 billion. It's a lot of money. You could give a lot of tuition scholarships for that.
Is that per year?
That's per year. Yeah. And of course, that's not the only cost of diabetes. There's the lost work days and things like that. So the total annual cost, more in the order of $100 billion. So now we're getting to your question about what happens after a meal. There is a condition called prediabetes. And prediabetes is when there is still some response to glucose, but it is not as full a response as in a normal individual. And what this is showing you is the insulin response in this case, this is a type 2 diabetic, an insulin response in someone whose blood sugar control is normal. This is someone who has impaired glucose tolerance, and here is a frank diabetic. So if you look at the timeline here, you're eating your meal for 15 minutes or so. I hope you're sitting and talking with friends. And then you watch your blood sugar rise. It'll be rising for the next couple of hours. Depending on what you eat, we'll get to that. And then it'll start to fall. If you notice, it's a little lower at that point than it started. That depends again on what you've been eating. If you have impaired glucose tolerance, it can be nearly twice as far before it comes down, and diabetes higher still. So we can detect these conditions metabolically by looking at blood sugar with time. This is just looking at that in a little more detail. It changes how quickly. That first graph made it look like it all happens at the same time. This is in a little more detail. Someone who has a normal glucose response will have their peak response of insulin. This is now releasing the hormone at 60 minutes after. Part of the impaired glucose tolerance is it takes longer to get that insulin into the bloodstream, so the sugar levels rise higher. And in type 2 diabetics, longer still. So the longer it takes to get the insulin out, the more chance there is for the sugar from your digestive process to accumulate in the blood. And this is showing you a day of eating. So we're starting here from 8 a.m. to 4 p.m. through 2 meals, not 3. And here's someone who has normal glycemic behavior, so a bit of a peak with breakfast and then down, perhaps below even the postprandial level. And then another peak after lunch and decline towards that fatal 4 o'clock where we are right now. I admire you for being so awake. In someone with impaired glucose tolerance, it will go high, but it will eventually get back down. It'll go high and come back down, sometimes not quite as far as fast. Someone with frank diabetes, it's actually going to never get down into the normal range. It will get higher and then it get not so high and get higher and not so high. So this is detectable as an abnormality before anything else in someone who is in the process of developing diabetes. Now, part of what makes this tricky is there are actually 2 phases of insulin response in response to a meal. If you look at a normal person, you have the meal, there's an early phase of insulin response that's independent of blood glucose, basically, very close to it. It's a phase that is related to the act of eating, almost. And then there's a second phase of insulin release that is, fine-tuning it. But this early phase here is sort of capturing the early part of it to keep it from getting out of control, and then the second phase can further manage it and spread it out over time. In someone who has type 2 diabetes, that early phase is not there. And so there is not the ability to keep it under control from early on. And therefore it takes that much longer to clear it out of the blood. This abnormality of not having the first phase of insulin response is an inherent trait, and it's independent of how healthy you are at the moment. I happen to have it. My mother had it. She also had diabetes. I'm trying hard not to have diabetes. It's pretty common. About a quarter of the population has this phase missing. And if it's missing, you're going to be at elevated risk for type 2 diabetes. And we'll see why that would ever get that frequent if we get to there. So this is kind of interesting. It's showing you that you spend, how much of the day you spend in the postprandial state with elevated blood sugar, right? And it depends on the time of the day, and it depends on the size of the meal. So the earlier you eat food, the quicker you return to a resting state. The smaller the meal you eat, the earlier you return to the resting state. So the rational thing to do would be to eat a large breakfast, a middle-sized lunch, and a small dinner. And of course, we do just the opposite. So if you eat a large dinner at 7 p.m., it's 4 to 5 hours before you get it back into the normal range. Not all foods are created alike. 2 dishes can have the exact same amount of glucose in them and have radically different effects on your blood sugar. And this is what we call glycemic index. How likely is it that this food will influence blood sugar? If you have a high glycemic index, you'll get a high blood sugar out of it. If you have a low glycemic index, you'll get a low one. Take a look at this. So here's new potatoes. Nice rapid rise. Actually a little undershoot, I think, there. Here's kidney beans. You can barely tell. And what it's doing is it's releasing that sugar. That sugar isn't getting thrown away. It's being doled out little bit by little bit so that it never rises into the overstocked state there. So these two have radically different patterns of being absorbed by the body. And this is showing that, sorry about the graphics here, I couldn't find a better one on this one, that the form of the food can affect glycemic index. So this is comparing cracked wheat, coarse flour, and fine white flour. And cracked wheat produces an elevation, but then it not a terrifically high one, and it gets back under control sooner. Coarse flour gets higher and stays up longer, and white flour is higher still. So the cracked wheat is almost not quite as benign as the beans, whereas the white flour is almost the same as the new potatoes. As a general rule of thumb, the darker it is, the lower the glycemic index. Things that are pale white tend to have a high glycemic index. People have become concerned about all this and have tried to find alternatives to sugar. So you're going to see a lot of things that have other things than table sugar to sweeten them, and they have different glycemic indices. So sucrose has a glycemic index of 70. That is, it has 70% the effect of pure glucose. Of course, it's half fructose, right? So that's not too surprising. Fructose has fairly little effect on the glycemic index, but you may remember back that it escapes some of the regulations, so it has other problems with it. Here's xylitol. This is a cute little, I think, 5-carbon sugar from trees. Almost no effect on blood glucose at all. And here's maltitol. If you've been looking for sugar-free candies, read the label. If it says maltitol, this is what you're getting. You're getting essentially the same blood sugar response as using sucrose. They are sugar alcohols, but they happen to be ones that the body can metabolize really fast. So maltitol is essentially useless as a way to reduce glycemic index. If I were to tell you how to improve the glycemic index of your food in 3 words, it's eat colorful food. If your plate is full of colors, it probably has a low glycemic index. And concern for this kind of thing is now widespread across the world, and different countries are developing suggestions that reflect the local culture so that you can eat things you like and still have a low glycemic index. And here's Canada. They obviously like whole grains a bunch. This really doesn't look that different from what you might see here, although the fishes may be more conspicuous. Here's the American pyramid. They fought tooth and nail. The food industry fought this for years because it was saying, don't eat too much fat, don't eat too much sugar. They fought that for years. This is a triumph of public policy. It may not look like it, but it is. Here's the one from Mexico. I think I like that one. You can see all the beans here, not so much on the whole grain breads, and lots of good veg fruits there. Here's Germany, another set of choices, and even China's developed one. So it's possible for anyone from any tradition to have good food that has a low glycemic index. And it is 4 o'clock, I think. If people want to stay for the last little bit, you're welcome to stay.